Hi everyone and welcome back to Advanced Higher Biology. Today we are on to Unit 3, Investigative Biology, and the first key area, Scientific Principles and Process. Now, this is a short unit, it's only 3 key areas, and something I've mentioned before in previous videos, some schools will do this unit first, as it's quite quick, it's quite short, and it gives you a bit of a underlying knowledge of the scientific method that you can apply to your project. Other people, this might be the last part you're doing. So depending on where you are, make sure you've watched the previous videos and hopefully it all goes in the right order for you. As with other videos, this is going to be slightly different from the advanced higher videos. The key areas are a bit shorter, so I'm not going to split this up into sub-key areas, and hopefully that'll make this a little bit quicker as well. So we'll just get through key area 1 today, and the next video will be key area 2, and so on. So, as I mentioned, there are 3 key areas in this unit, and they all revolve around investigative biology. The idea behind this unit is looking at how you go through designing and carrying out a scientific experiment or an investigation, and how you would critically evaluate this. Why are there things that go wrong? Why are there some things that are unreliable? You might remember the scientific literacy part of National 5 and Higher. You had to talk about things like reliability or validity. This just goes into a little bit more detail about this. Also some of the ethics revolved around experimentation, also how you would share all this data as well. And it gives you a grounding in just the real world science, the application of science, and how this actually all works. So, as I said, we're going to start off with scientific principles and process first of all. And the first area of this is something called the scientific method. This might be something you've came across before when you've been designing experiments in previous areas of school. So, first of all, the scientific cycle or the scientific method is a set process. So all scientists have to cover this. Whether you are doing an S3 experiment on transpiration or whether you're completing your PhD, you still need to go through an idea, you need to hypothesize, you need to research and carry out an experiment in a certain way. Now, one of the ways to summarize this is this cycle here. It's quite common for this to come up as almost a multiple choice question or to ask what would be the next stage. So it is quite important that you know the order of this cycle. Some would actually argue as well, it is more like a spiral because although it continually goes in a cycle, you also should be moving upwards as you gain more knowledge as well. So essentially what should happen is you should have some form of observation. You're looking at something and you think, what would X do to Y? And this would be the idea behind any scientific experiment, is you're looking at something and wondering what would happen. As you have came across before as well, from that you need to then figure out a hypothesis. Your hypothesis is a theory, it's what you think will happen, and we'll go into more detail about a hypothesis shortly. Through the next part, experimental design, it's exactly as it says. You would then design this experiment. You would work out how you would carry this out. What are the variables? A lot of detail goes into this design, and that is part of what this unit and also your project will be about. How do you actually set up a valid, reliable experiment? Once you've actually carried out an experiment, then you're going to gather the data from it, so find out what has happened. You might be taking readings, you might be measuring things, you might be timing things, but you gather your information, you record it, and then you're going to analyse that data. When you analyse that data, and again, this could be creating graphs, it could be looking at statistics, it could just be comparing data sets to see if there's any differences between these different variables. But that main analysis part is when you're actually looking at what has happened. Is there any effect or is everything all the same? From that, you're then obviously going to evaluate these results. So you're going to decide, as I increased X, then there was a significant difference in Y. You would look at what has happened. Can you justify this? Is there any missing pieces here? And obviously that will lead to your conclusion. And your conclusion, coming full circle, will be what happened to X as you increased Y, for example. How this is a cycle, though, is what then might happen is you might have further ideas. How can I go and find out more about this? Or is there more detail I can go into? Is there more variables I can use? Were the concentrations too far apart? Now I need to delve in and make them slightly different each time. And this is where this whole thing will go around. The process of science is never finished. You're really just starting off with the best idea at the time, and you can always refine that. You can expand it and find more information. So you may revise that hypothesis if you think, right, well, what is the difference now, and continue this cycle. But again, Important for you to know what these all mean, but important for you to know the order of these in particular. Now, like I said, the idea of all this is that there's always a lot of debate about what is true and what is not in science, and that's kind of the point of science in general. It is very normal to refine ideas. Some incredible scientific breakthroughs in the 1800s are now obviously very much questioned, and we look at how we can then go and make this a better scientific investigation, refine methods, all these sort of things. So any scientific theory is really just regarded as the best explanation to date. It might be absolutely correct, but it might be what people thought at that time or with the tools available, and if there's further evidence, then that will be updated. Now, I talked about hypotheses earlier on, and I did mention that this all comes into a bit more detail in National 5, in Advanced Higher. Now, first of all, a term you'll come across is the null hypothesis. Now, a null hypothesis is basically a statement that says there will be no effect as a result of your treatment. So, for example, if you're looking at a plant and your hypothesis is increasing the temperature will increase the transpiration rate, your null hypothesis would be that temperature has no effect on transpiration rate. Now, sometimes these questions can be really annoying because often people say, in this case, temperature will not increase transpiration rate. That's still a statement, okay? Whereas the saying there will be no effect is more essentially vague. Your point in a scientific experiment is actually to disprove the null hypothesis. If you have a statistically significant increase or decrease due to the variable you've used, due to the treatment you've used, then what's then going to happen is you're able to reject the null hypothesis because you can say there has been a difference. So therefore there has been an effect, therefore the null hypothesis is wrong, and ideally your hypothesis is right. So null hypothesis, there will be no effect due to your treatment. Another thing that people always get mixed up about is, even in school, if you do not have a result from your experiment, if you find nothing out, if there has been no effect, that is still a valid finding. That is still something new that people maybe did not know. So often people think if, again, going back to the temperature example, if you found there was no effect on increasing temperature on transpiration rate, that's still a finding, and that's still a valid finding, as long as your investigation is well designed. If your investigation is not well designed, if there's a lot of confounding variables that have messed things up, if you've only done it once and not repeated it, a lot of other factors as well, then no one will really bother about this because there's no proof that this was a well-designed experiment. However, if it is well designed, a negative result is still a valid finding. And in fact, just using an example here, there is actually a journal, an academic journal, that literally just uses these negative results because they are all still very scientifically relevant. Now, when we ever have a conclusion from a scientific experiment or an investigation, if there's any conflicting data, if there's two studies and they're both saying totally different things, what will normally happen is you'll go through a careful evaluation to see if there's any errors, any mistakes, anything that could have went wrong in the scientific investigation. But usually this will actually lead to further experimentation. Okay, like anything else, right, we've got different results, so we're going to do it again, we're going to keep doing it until we find out something else. It's a bit like when you did reliable results in, say, National 5. You may have one data set that's a bit wild, so therefore you're going to have to go back and actually go and try again and keep going to see what is the true result. When we talk about validity, again in Advanced Higher, validity is something you've came across in higher biology. This is, again, really important, especially when you're designing your own project. But in order for an experiment to be valid, you will need to be able to reproduce these. If you just have a one-off data set and you say, oh yes, because I increased temperature, transpiration increased, that's not good enough. Okay, there needs to be more to this. And again, we'll go through a lot more detail about how you actually set up your experiment. Now, one thing you may use in your project, or you may not, is the use of statistics. If you go into any form of science in university, it's one of the first things you'll be dealing with. Rather than just looking at this number is different from this number, you're going to use statistics to say how likely it is that these differences are just due to chance. And that's really the whole idea. Statistics looks really, really scary, and you'll come across p-values of 0.05 and think, what's going on here? Really, all you're doing with statistics is you're proving or trying to show that the results you have found are not due to chance, so not just due to a random observation. And that goes back to looking at your null hypothesis. So, for example, if you have a result and you find statistically there's no significant difference between those two data sets, then the difference could just be due to chance. If you find there is a significant difference, and this is again something you look at when you look at error bars in graphs, if they're like overlapping, then there's no significant difference between them, and it's very difficult to actually say those data sets are different, and therefore there was a difference of the treatment and there is a statistical impact. If you're looking at one data set that is absolutely massive on one side with a tiny error bar, there's no range whatsoever, and the second treatment is the opposite, it's tiny, and again with a tiny error bar as well, then you can say, using statistics, these results are not caused by chance, we reject the null hypothesis, and my initial hypothesis is correct. But again, this is something you may use or you may not, but it's good for you just to know what we use. And as I mentioned, that's then going to have an impact on your null hypothesis. So if there is no statistically significant effect, then your null hypothesis is going to be found true. Just again covering this part here as well, okay, this is if you do get a statistically significant difference between treatments, then you reject your null hypothesis. One of the last things we look at here in this part of scientific ideas is if someone produces a scientific paper or comes up with a new theory, discovery, any information whatsoever, you can't then just go and share that information and say, this is what I found. There needs to be a process to make sure that this is all valid, that it's all significant. So all of these scientific ideas have to be checked independently. And this is part of what we go into in this next sub-key area here. So again, I am just going in for this whole key area in one go. It's quite dry. There's not a lot of fun information in Unit 3. So you might want to go and pause it and come back to this at a later point. Or if you're just hearing through it, then excellent. We'll just continue on. This one's a lot shorter. So in terms of having all that information checked and shared, one of the things we look at in this unit is how we actually communicate scientific findings. Now, often what we do is this goes in the form of journals. This might be something you've came across before. Journals are scientific articles that are published to various journal companies that then go and share these out. You'll find some of them online, some of them are free, some of them are very expensive, which leads to a bigger debate as well. But that is then sharing your knowledge as well. The other things we can do as well is go and share these at conferences, at lectures. The whole idea is you're sharing your science with other experts, with other scientists that are able to look at this and discuss. And one of the really important things is that when you share a journal, which you'll look at in a bit more detail, probably when you're doing your project, is that you need to have your method, your data, your analysis, your conclusion. You need to have all your information available, laid out, especially as well if you potentially have had funding from a group. That's a big issue and controversy in science. If you're disproving or trying to disprove climate change, but you've been funded by a fossil fuel company, then you have to declare that. If you don't declare that, then that looks a bit dodgy and people are not going to listen to you. So like I said, these all go through academic journals. There's also these talks at seminars. Posters are another one as well. You have academic posters, which are literally just a summary of your journal, usually on a big A3 page. And again, you can have these conferences where everyone has their posters and you go across and you debate with people and you say, oh, that's interesting, I've been working on this as well. What were your thoughts? How did you carry this out? Oh, wow, did you think of this? Again, science is meant to be open, it's meant to be debated. It's not just one person in a lab saying, this is the correct result, this is the correct answer, and we move on. What we will do is in key area 3, I believe, we'll go through journal articles and actually look at how to look at these. If this is you starting to look at advanced higher biology, one of the things I would encourage you to do is go and just look up some journal articles as well. Some of them, they might just look like gibberish to you. You might think, I don't know what this actually means, but it's good to try and build up your scientific literacy and build up some knowledge of what's going on, how this is shared, what the issues are, how people carry out various investigations. It'll open your mind to a lot more ideas and also how these processes all go across. If you just Google these or even go and Google Scholar, you'll find a lot of free PDFs as well, so rather than spend a fortune on subscriptions. But again, these articles are all current, up-to-date knowledge of really the forefront of science and how people are carrying these out, how they're going through the scientific methods, and what their conclusions are. Now, the last part here, and again, this is a really common question, is looking at how that information is all checked. So we stopped the last key area talk about how information has to be checked independently. You can't just publish and then say, yes, this is true. People need to look at this. And this is a process called peer review. Now, peer review is when any information, any investigation or experiment is critically evaluated by specialists with expertise in the relevant field. Now, this bit here is really important because if you, for example, are an evolutionary biologist, you can't then just send your paper or have it checked or peer reviewed by someone, for example, who has a PhD in astrophysics. It's not relevant because they're not in the relevant field. The same would be the other way around. You need a body of people, usually 2 or 3 other experts in that science, who look at your information, check if it's valid, look for any maybe issues with the variables or reproduction or how you've actually carried out the experiment. Again, they will have knowledge or more knowledge of the methods used in this practice. Now, like I said, this is a really common question, and a lot of times people get it wrong. The reason why they get it wrong is they'll often say something like, oh, this is when scientific investigations or write-ups are evaluated by other scientists, and they won't mention this idea of experts. So experts in their field is just critical for you to write. So if there's one thing you remember from this unit, please remember the definition of peer review. It's critically evaluated by specialists with expertise in the relevant field. Now, one of the things that would hopefully be a good discussion in your classroom, and we're not going to go into a lot of detail in this video, is to look at the importance of sharing science, because it's important to share science with experts so everyone can evaluate this, so we can all have the best scientific processes possible. But it's also really important to share with the wider public as well. And it's one of the things that's become an issue when you look at all the sort of fake news and bad science. There's a lot of information that gets shared out, then someone will take the wrong idea of it basically, turn that into a headline, and then people who do not have scientific grounding understandably look at that and think, oh, what's going on? So very quickly there's a bunch of just totally unhinged headlines here from the last little while. Again, none of these are true in any possible way, but there's been a part of a study that someone's kind of misunderstood or exaggerated massively. If you go onto any sort of mainstream news site and delve into the science section, you probably will find something. Normally it's to do with one tiny investigation that's found something that potentially could help in a very specific form of treating cancer or something that has been blown up to be scientists find cure for cancer. You know, and it's just looking at this and saying it's leading to a large misunderstanding by the public, which leads also to distrust. And it's just a big thing that we need to all acknowledge and talk about, is the importance of science in the media and sending it out properly. Right, we're on to the last part then of scientific principles and processes. And again, if it's quite a lot for you, and I understand a lot of it isn't the most exciting, then have a pause, come back to it. But we're just covering all the main parts of Unit 3, Key Area 1. So this last part goes on to ethics. Now, this is something that you probably have some sort of background information about, but you've potentially not went through it before in class. The idea of the ethics of an investigation. And a lot of this should hopefully be quite common sense, but there's a lot of keywords and terms that you need to know for this part. So the idea is that no matter what we say in science, no matter how big the discussions and the controversies can be, the idea is that everything needs to be honest. There needs to be utmost integrity when you're sharing your information, when you're carrying out investigations. That's the key part of then sharing all our studies, all our data, and everyone can look at this. Now, in order to be honest, you're going to first of all have unbiased presentation of your results. You might go into your project, or you might go into a PhD or some form of academia later in life, and you desperately want one result. However, as a scientist, you cannot be biased in that. You know, you can't just lean into that and ignore other data that disproves it. You need to be unbiased in your presentation. You also need to provide references for all the work. And again, this is something we'll go into in a little bit more detail later on. but references and citations are a way of showing information from other people that you've gathered from, say, other academic texts. This might back up what you're saying, it might be another theory, but you should be going in, collating all the information around the area you're looking at, sharing that, but you have to reference it to show I got it from this person here. And as a side part of that as well, your main thing is avoiding plagiarism. You can't just steal someone else's work, you can't copy other people's work and pass it off as your own. It's the same in all walks of life. These are all just unethical things that you need to watch out and avoid. Now, again, another part we'll go into here is the use of animal studies, and there's something called the 3 Rs. Essentially, and again, this shouldn't be too surprising, but what we do now with any form of study involving an animal is we look at having this potential suffering of the animal kept to an absolute minimum, and there's different ways of doing this. Back in the day, science was maybe a little bit more unregulated, and there's a lot of controversy, a lot of things that shouldn't have happened. Now we're hopefully doing a much better job, and you need to always take into account the 3 Rs if you're designing an experiment with a living organism. Now, what I've done is I've tried to summarize these here. You just need to know them. You just need to learn the terms. Again, common question to be, oh, someone has used replacement in this animal study. What else could be used to keep animal suffering to a minimum? So basically, we have something called replacement, which, as the name suggests, you're going to replace the animal with an alternative. Now, the idea here is if you're using, for example, a mouse to try out a new drug, do you have to use a mouse? Do you have to jump to a mouse straight away? Is there a less sentient species? Or could you even just be using this in a tissue culture? If, for example, you're looking at the effect on muscle growth, could you just use muscle tissue and add this to it in a petri dish? And therefore you don't actually need to go and inject a mouse with something that we don't know what's going to happen. That would be a form of replacement, is sort of lowering the sort of hierarchy of the animal that you're using. The other is reduction. So again, you need to have robust, reliable results, but do you need to do this on 10,000 mice? Could you do it on 1,000 mice? Could you do it on 100? Again, the clue is in the name, but reduction, using the minimum number of animals to obtain robust results. So trying to keep, again, the suffering to a minimum. And finally, refinement. That is when you use methods that minimize the potential pain, suffering, or distress of the animal. And there's a lot of different ways that you can do this. But again, any form of looking at this and thinking, well, rather than using this method that's going to be very stressful and it's going to harm the mouse, is there any way we can use this? Could we have them knocked out? Could we use a smaller injection while we're already doing this part? This really comes into the scientific method as well, or scientific experimental design. Any way of making the experiment better to reduce harm would be refinement. Now, if you go on to humans as well, this is where things also get a bit more intense. There are a lot more concepts to ensure human rights are adhered to. So again, this starts to go into a bit more of a sort of moral dilemma. So, for example, the picture here, I believe, was a study on pollution, where the people who are sitting just writing how they're feeling and I think completing sort of various logical tasks, they're all just getting pumped with various gases that are full of things you shouldn't be breathing in. You know, technically, that's at that point it was scientific, but now we've got more ethics to say this is maybe not a good idea. So if you're in a situation where you're involving humans, there's a bunch of different things we need to look at, and a lot of it comes down to obviously your human rights, but also the fact that you need to know what's happening and you need to be able to get out if you want, and also your right to anonymity as well. So for here, you also need to justify why you're doing this. Now just strapping someone up and poisoning them isn't a good enough reason to carry out an experiment. In this case, which obviously is quite extreme, you'd be having to justify something like, oh, you live in a very, very high polluted area and we are using this to kind of show this is the impact of the pollution on these people, but also go through all the steps to ensure that all these participants are totally happy and understand the impact and more of that side of things. So first of all, you have informed consent. So like I mentioned, this is making sure that the participant is entirely aware of what's going on. So not only what's going to happen, what's going to be done to them, how it's going to happen, what are the potential risks of this as well. The idea here is this participant needs to be entirely aware of everything that can happen be happy to do it, and sign up. On the back of that, though, is the right to withdraw. So even if you have provided consent for this at any point at the trial, and this is, again, something that people tend to forget about, either in the day leading up to doing this trial or during the trial itself, at any stage at all, the human participant has the right to say, nope, I'm out, I'm not happy about this. There can be no coercion, there could be no, oh, but you've signed this. It's entirely within your right to withdraw at any stage. And finally, the idea of confidentiality as well. So data cannot be shared without this person's consent. That both revolves around not using their name, giving themselves potentially, say, a randomly allocated number or letter. So we know it, you'll come across things like patient N did this. They're not just saying John Smith experienced this or John Smith from this area of the UK. So any data collected cannot identify the subject. They have the right to choose to remain anonymous. And again, like everything else, data cannot be shared without their consent. Now, because of that, in terms of human studies, we have a lot of legislation and laws, and these are here to govern various aspects of science. The whole idea for this is, as you've seen throughout this key, and this is the end of it now, there's a lot of things to think about when you're carrying out some form of scientific method, but it is so important that it's not misused. That's why we now have the 3 R's for animals, we have human rights for humans, but we now have laws as well. You can't just go and do certain things. These laws are different in different countries, sometimes with different beliefs or ethics or morals. But again, you need to be very aware of the laws, and hopefully there's nothing you're doing in an advanced hire project that's going to trespass any of these. But we now have legislation to stop or limit the potential for the misuse of scientific studies. So that is all for this key area. So again, we went through the whole thing. That is 1/3 of Unit 3 done. It's quite heavy. There's a lot of words. There's a lot of terms that you just need to know. But more often, it's also for you to think about for your processes of science, design your project, and even the sort of experimental design questions that you'll come across in advanced higher biology. The idea is, rather than just sort of regurgitating terms, you need to show a bit more of understanding in advanced higher. So if you're looking at any information to do with studies, have a think about, did they carry out the 3 R's? Did that human have the right to withdraw? Was there an issue with this? How was it studied? Did the person share the information? If so, how? And that's the main parts for Unit 3 Key Area 1. Excuse me. So that is all for this key area. We'll be back with key area 2, experimentation. Hopefully you found this quite useful, and regardless of what stage you are with your advanced hire, hopefully you can also use this when you're planning out your projects and things as well. So that's all for this, and I'll see you next for key area 2. Bye for now.